So I welcome you all to this international conference on advanced materials in innovative technology, MET 2022. So today, as a session chair, we have Dr. Manali Datta. She is a structural biophysicist who has dealt with the functional dynamics of proteins as a part of doctoral and postdoctoral studies from IIT Roorkee. She focuses on the development of protein interaction-based diagnostic for lifestyle disorders and microbial disease. She has also got 33 journal publications and 8 book chapters, and she has 5 patents in the IPO. I welcome Dr. Manali.
Thank you, Dr. Pankaj. It is a pleasure hosting this session. So participants will begin with the session today. We have the first participant, Mr. Shobhit. Mr. Shobhit. Okay, the next participant, Ms. Anushka Tyagi. May I please request you to share your screen?
Good morning, ma'am.
Good morning.
Good morning, sir.
Good morning.
Ma'am, may I start?
Yes, please do.
Okay. My screen is visible, ma'am. Am I audible to you all?
Yes, yes, you are.
Okay, ma'am. Good morning, all of you. Myself, Anushka Tyagi, research scholar from Jyoti Vidyapeeth Women's University, Jaipur, Rajasthan. My title of research paper is Synthesis and Characterization of Swarn-Makshik Bhasma. My present work comprises of introduction, experimental techniques, result, and last one is conclusion. Introduction. Metallic preparation known as bhasma, fine powders, which are fine powders obtained through calcination, are unique to the Ayurvedic system of medicine in which the metallic state is transformed into the suitable compound formed with the therapeutic activity. Swarn-Makshik Bhasma are claimed to be biologically produced nanoparticles which are prescribed with several other medicines of Ayurveda. My present study examined the utility of Swarn-Makshik Bhasma along with Tulsi extract for the treatment of anemia, which is mainly iron-based. The concept of using nanometal particle is prevailing since Charak Samhita, which is the ancient Ayurvedic text. Swarn-Makshik Bhasma comes under the group of chalcopyrite having huge therapeutic value. Swarn-Makshik Bhasma is an copper and iron-based formulation, has been used in therapeutics of anemia, jaundice, poor digestion, edema, skin disease, and in so many other diseases. This paper aims at providing specific good manufacturing procedure, GMP, for the manufacture of nanoparticle of chalcopyrite ash, and also attempts to study the effects of different steps and process on copper iron sulfide ash formation. The factors affected by side reduction, bulk versus nanomaterial bhasma drugs. These are melting points, optical properties, colors, surface reactivity, magnetic properties, conductivity, and bioavailability. Experimental and characterization techniques. Synthesis of Swarn-Makshik Bhasma. The second one is structural characterization, XRD, X-ray diffraction method. Third one is morphological characterization, SAM, scanning electron microscopy. Preparation and nanotization of Swarn-Makshik Bhasma. First of all, we purchased the raw material for Swarn-Makshik Bhasma. Then Swarn-Makshik Bhasma powdered, Swarn-Makshik powdered and sieved. Then powder placed in an iron pan and Nimbu Swaras is added and heated. Stirred continuously till the moisture has evaporated. Then shoot the Swarn-Makshik titrate with Nimbu Swaras in kharal. Then mixed and dried. Cakes kept in syrup samput. Give Varaputta 10 times, which is mentioned in ancient Ayurvedic text. The procedure is mentioned. Cool temperature, at room temperature, and then collect the cakes, grind in kharal and make fine powders. Then we prepared nanomaterial. 500 mg of bhasma was taken and 5 ml of MQ water was added. Mixed with water and suspension was kept static to settle down bigger size particle for 2.5 minutes. Then finer suspension from top was taken for downstream experiments. Then concentration of particles and characterization will be done, and the other side used for in vitro experiments. Structural characterization, XRD experiment, X-ray diffraction method. The XRD pattern of Solmakshi Bhasma as crystalline material with decreased particle size. The peaks are shown in picture. The presence of sharp diffraction peak shows the highly crystalline nature of Solmakshi Bhasma. The sample shows diffraction peak majorly at 2 theta is at 34.50. 36.1, 41.36, 49.16, and 54.32, 62.5, and 64.58, as you can see in this image of XRD in finally prepared Bhasma, which indicates the planes with reference to JCP-DS file number confirming the presence of chalcopyrite. The XRD pattern in final product shows that iron oxide is mainly present in form of FeS2, iron disulfide, and copper iron sulfide during heating in Bhasmikaran at higher temperature 650 degrees Celsius. Chalcopyrite and iron oxide formed in its most stable form. In this, copper iron sulfide in mixture of two states of copper and iron. Morphological and optical characterization. This is the image after the SAM study, scanning electron microscope. The study revealed regular and uniform arrangement of clusters of granules in finally prepared bhasma, which was not observed in raw material. It is clearly observed that the surface area was smooth after final bhasmikaran. In the presence of plant extract Solmakshi Bhasma formation, the particle size is small as revealed by SAM studies. The surface energy of surface precipitate Solmakshi Bhasma would be dominating the shape of the crystals, and therefore the spherical crystallites are observed, as you can see in this image, which is the image of SAM. The conclusions of this study are the following significant achievements have resulted during the course of our studies. All the analysis of scanning electron microscopy and X-ray diffraction analysis confirm the formation of chalcopyrite nanoparticles. The self-organized Bhasma nanoparticles thin film gave rise to another interesting feature with the smooth surface clusters, and the average smallest cluster size was predicted around 200 nanometer. The X-ray diffraction analysis further confirmed the lowest particle size of nanoparticles is around 95 nanometer. These are some references which I have mentioned. Thank you.
Thank you, Anushka. Very concise and well presented data.
Thank you, ma'am.
Any questions from the participants? Okay, in that case, Anushka, the first thing I would like to ask, when you're preparing this bhasma, what is your ultimate goal?
Sorry, ma'am. What's my ultimate goal?
Goal, yes.
Yes, ma'am. Actually, I want to examine the actual particle size, minimum particle size, nanoparticle size, which I have examined through XRD and SAM study. So मुझे exactly पता करना था कि इसमें iron oxide, copper iron sulfide present है कि नहीं है. Or size, mainly size. What is the size of these particles?
And when you are saying that it is being used in Ayurveda,
Yes, ma'am.
detection of the size or the we can say the particle size, what validity will it have in the Ayurvedic application? Do you think any implications are there? अगर साइज छोटा है तो अच्छा काम करेगा, साइज बड़ा है तो कम काम करेगा, कुछ ऐसा कोरिलेशन है?
Yes ma'am, exactly. अगर साइज छोटा है तो मैम वो इजिली सेलुलर अपटेक इजिली हो जाएगा.
Okay. In the protocol, you have exposed this bhasma, Swarnamakshika bhasma to, I think, Nimburas, or you have used a different name for it. So what was the significance of that?
मैम सरफेस पैसिविटेट जो होता है उसमें इजिली वो हो जाता है, कैपिंग हो जाती है सरफेस की, तो जिससे साइज रिडक्ट करने में रिड्यूस करने में हेल्प होती है.
Capping has a different function. Okay, when you are using lemon juice, what is the actual component which is helping in the capping of the nano bhasma या जो भी आप इसको नाम दे रहे, which is the component? Have you done the FTI?
No ma'am.
Okay, any idea?
No ma'am.
Okay, it is citric acid basically. Okay, thank you Anushka.
Okay ma'am, thank you so much.
Thank you. May I now request the next participant? Mr. Shobit, are you available now?
Yes, ma'am. I am available.
Yes. Please.
Am I audible?
Yes, you are audible. But
Okay. Very fine. Very fine. Let me share my screen. Is it visible, ma'am?
Not on our end.
Hello.
Not on our end.
Now it's okay.
No.
Is it now visible?
Yes. Okay, just press F5.
Now
F5 have you pressed?
I pressed.
But Shobit, put it in this full screen mode. Go to this top button which is there, slideshow. Drop down menu would be there. Home, insert, design, animation, slideshow.
I done that, sir. I just.
But still it is not in that mode.
Still not now.
No.
I just press it. From where are you saying? From insert.
This on the top, slideshow. Come to the right part, design. Right. Move to right side. Yes, animation. Now slideshow.
Okay, fine.
You start.
Actually, I was doing from
the beginning. From the beginning.
It's visible now.
I think there is some lag.
Okay. Actually, it's showing in my PC. It's not a problem, but I don't know what's happening here. How much is it visible?
The fonts are very small. That's the
Fonts
thing.
are very small.
Me as well as the other participant won't be able to. That's the
I enlarged it, sir. It is in slideshow mode. It is visible to me.
But it won't help us, right, Shobit? The presentation you are making to us, not for
Ma'am, let me verbally demonstrate and let me finish, please.
us. Otherwise, please go ahead.
I will wrap it up. So I will speak slowly so that everybody will make it understand. Na?
You have
Okay.
10 minutes. Please.
Okay. Fine.
Okay.
Good morning, everyone. I, Shobit, PhD scholar, Department of Food Technology, would like to present before you all my presentation topic entitled Effect of Pre-Treatment and Fluidized Bed Drying on Rehydration Studies of Moringa oleifera Leaves. Okay. Let me introduce you about the drumsticks. Actually, what is drumsticks? What's their importance and their origin and the production and some nutritional as well as medicinal property which Moringa oleifera leaves basically possesses. Okay. So drumstick, which is also known as Sahajan, Sohanjna and horseradish tree, generally belongs to the family Moringaceae. It is a fast-growing, drought-tolerant, and exceptionally nutritious tree, and almost its every part can be utilized for food or some other beneficial application. That's why it is considered as a superfood or miracle tree. And it is native to India basically, but it is also cultivated in countries like Bangladesh, Afghanistan, some West Asian countries, East Asian countries, Arabian Peninsula, some African countries such as Ghana, Nigeria, Rwanda, Kenya, Mozambique, South and Central America regions, Mexico, Paraguay, Peru, and some part of Brazil. Okay, so India is the largest producer with an area about 0.4 lakhs hectare, and generally produces about 80% of the world Moringa oleifera leaves. In India, major producer states are Andhra Pradesh, Karnataka, Tamil Nadu, Odisha. India is generally a core supplier of Moringa and generally fetching crores of rupees as foreign exchange due to this. And global market, as far as global market is concerned, India is likely to cross around about 7 billion US dollar by 2022, and with a compound annual growth rate of 9.5 per year. And Moringa oleifera leaves basically contains high amount of protein, fiber, fats, minerals. Among minerals, majorly calcium, magnesium, phosphorus, potassium, copper, iron, and zinc. Whereas it has also several rich in a few of the vitamins, especially vitamin A. It is which has precursor beta carotene, and vitamin C also. So Moringa oleifera leaves generally has 9 times of protein than of yogurt, 10 times vitamin A of carrot, 17 times of calcium of milk, 15 times potassium of banana, and 25 times of iron of spinach. Okay, so its leaves are also rich in several other biotic compounds, generally essential amino acids, several sort of phytochemicals like cytokinins, sterols, alkaloids, flavonoids, tannins, saponins, phenolic and glycosidic compounds. Especially it has a broad range of flavonoids as well as for polyphenol compound. Among polyphenols, it mainly constitutes gallic acid, salicylic acid, genestic acid, allergic acid, ferulic acid, caffeic acid, picomeric acid, and chlorogenic acid. Whereas flavonoids includes your quercetin, camphorol, isoquercetin, rutin, epicatechin, catechin, lutein, genetin, and tyrosine, and myricetin. So these are the compounds which makes it more suitable for consumption, as it has lots of medicinal property due to that. Okay, so it can cure around 300 diseases, such as it is reported that it can cure around 300 diseases like your anemia, asthma, arthritis, rheumatism, cancer, constipation, diarrhea, diabetes, headache, heart problems, kidney stones, stomach and intestinal ulcers, stomach pain, intestinal spasm, thyroid disorder, bacteria, fungal, parasitic, and several viral disease also. So blanching, I performed some pre-treatments for this study, and among that, blanching was a basic pre-treatment which I performed. Blanching is basically a process to scald a food commodity in boiling water for a very short period of time, and then plug into hot water simultaneously so as to avoid over blanching. It is basically helps in preservation of flavor, color, texture, retention of nutritional value, protection of various bioactive ingredients from drying effect, and also removes dust, dirt, microbes, and other organisms from the surface of leaves. Blanching basically inactivates, the basic goal of blanching is to inactivate enzymes, especially lipoxygenase, polyphenol oxidase, polyglycerurinase, chlorophyllase, which can cause deterioration of food compounds. Thus, blanching helps in reducing quality loss over time. So after that, after these pre-treatments, I performed several type of, I performed a fluidized bed drying technique. So we will have a look at the methodology now. So let me give you some idea about what sort of methods I opted. So as you will be able to see this that I performed 6 loads were there for blanching. I have 6 loads. In the first group, only a blanch sample, that is basic blanching, hot water blanching at 85 degrees Celsius for 60 seconds was done. And in the second group, blanching with a chemical treatment, that is basically a preservative, and that is KMS, potassium metabisulfite, was used. And the leaves were blanched at same time temperature combination at 85 degrees Celsius for 60 minutes. And in the third load, blanching along with sodium bisulfite was used, and the time temperature combination was 85 degrees Celsius for 60 seconds. In the fourth group also, blanching with sodium bicarbonate, and in the fifth group, blanching with sodium chloride. And then in the last load, only leaves were dried without any sort of blanching, so as to compare the impact of blanching or drying alone. Then after performing these pre-treatments before drying, I performed drying technique, that is basically fluidized bed drying. I opted for fluidized bed drying, and in that I have 3 different temperatures, that is 50, 60 and 70 degrees Celsius, with an air velocity that is fixed. 10 meter per second air velocity was given to the sample source. And after performing this, drying was conducted until we obtained constant weight. And after that, samples were transferred and labeled in a transparent cellophane bag, and it was kept in desiccator for further sort of analysis. So basically, these are the rehydration studies which I performed after applying the several type of pre-treatments and after following drying techniques. So basically, I studied some rehydration studies of the 6 treatments which I generated. So among that, one is a rehydration ratio. It's a major and most important parameter of rehydration studies, and it is given by weight of drained sample divided by your weight of dried sample. Whereas relative reconstitution capacity, it is generally weight of rehydrated greens divided by weight of your fresh greens into 100. Dehydration ratio, it is basically weight of dehydrated leaves divided by your initial weight of the sample. What sort of initial weight you firstly taken, that will help in governing dehydration ratio. Whereas coefficient of rehydration is the ratio between rehydration ratio as well as your denominator, it is dehydration ratio. So the ratio of both rehydration as well as dehydration will give you some idea about the coefficient of rehydration. So these were the methods which I followed for pursuing rehydration studies, and these were the observations. And it was observed that sample which was blanched with KMS has higher rehydration ratio as well as dehydration ratio at 70 degrees Celsius KMS treatment, as you can clearly see with the graphs. So as far as relative reconstitution capacity is concerned, it was observed that fluidized bed drying 60 degrees Celsius was found superior among all the treatments, whereas in the coefficient of rehydration, as far as it is concerned, that it was also found that fluidized bed drying at 60 degrees Celsius was found superior. So in nutshell, I would like to conclude that rehydration ratio and other properties were found at which temperature poses higher, which was found at higher temperature basically. So high rehydration ratio means dried product has good quality because because the pores will absorb maximum amount of water to enter the cells. It will have a higher tendency because structure will get open, and and that's why water holding capacity will increase. And it can also be said that it can also be said that higher the porosity, better will be faster will be the rehydration, and better will be the rehydration, and better will be the output product. So these were the properties. As far as alone drying sample is concerned, it was not so the case with the drying without any blanching treatment, because of the reduction of several type of capillaries, that is capillaries got shrunken, and leaded to reduced hydrophilic properties, so that water was not able to imbibe enough, so that sample source can fully hydrate. That's why the treatment which have only drying, that processing step has less rehydration ratios, as well as relative reconstitution capacity, as compared to those which were earlier pre-treated. So that was all about my findings. Thank you. Hello.
Thank you, Shobit. It was a concise presentation.
Thank you,
Any
ma'am.
questions from the participants? Fine, Shobit. Then I'll ask questions from my end only.
Yes, ma'am.
You use 6 chemical additives.
Please speak loudly, ma'am.
You have used 6 chemical additives.
Six pre-treatments. Yeah. One is blanching plus KMS. That is basically
I saw. According to you, which additive is acting the best out of all them?
KMS, blanching plus KMS combination was found to be superior among all the treatments, as it leads to better rehydration, and other than though results were not that much variation was there, but if you compare and you see that blanching plus KMS gave me the best results.
In the reconstitution ratio,
Let me come to this one.
I noticed that the control for FBD 60 is giving a better value than all of the FBD 70. Can you just switch to the slide?
This one?
Second last slide.
Second last. This one, rehydration.
Constitution.
Relative reconstitution. You are talking about relative reconstitution?
Yes.
RRC?
Yes.
Okay, this one.
It's not flipping on my end, but as far
as let me get your question. What you are trying to say?
I am trying to say that if you notice the control value for FBD 60.
Yeah, it is showing better results. Yeah.
So why not go ahead with that without adding any additives, chemical additives?
No. It is just a trend with this technique, and now the variation is because you are not able to see not that sort of variation. It is changing in fractions at third decimal place. So in a nutshell, I concluded so it's not that all the trends are of KMS and all that. So there is a difference that it is mostly it is found that blanch plus KMS is showing the better trend, but variation, whether it is wheresoever variation is there, it is on the third decimal place after like 0.538, something like 0.532. So something variation is not that much extreme. So I let me finish, ma'am. So I dragged a conclusion that in most of the parameters, blanching plus KMS is showing the better results than the other. And one more logic behind that I found, because I opted that with blanch plus KMS is highest, because it will lead to because it is a preservative and it will retain maximum sort of nutritional as well as some sort of biotic compounds retention will be there. Moreover, I have chosen I have some other studies also. So keeping in view of nutritional as well as medicinal as well as phytochemicals, as well as several sort of medicinal properties. From each and every aspect, I just opted that KMS is showing much better trend among all the samples. And moreover, it is also preventing a cellular rupture, because it will because if there is no chemical treatment and something like that, so our cell wall might be possibility that it will rupture and it will lead to more losses. So in control, it's a loophole, I think.
Fine. Thank you, Shobit. You can
Okay.
answer this please. The next
Oh,
participant,
okay, ma'am. Thank you so much.
Mr. Sheikh Adil.
Yes, ma'am. Yes. I'm
Yes.
okay. Actually, I'm facing problem. You cannot start screen share while the other
So yes,
participant.
Shobit, please.
I'm doing stop sharing. I'm doing, yeah. I think it is done.
Okay. Ma'am, is my screen is visible to you?
Just press F5.
Yes, yes, ma'am.
Yeah,
Okay.
yeah, perfectly.
First of all, good morning to all, and thanks to coordinator as well as management to give me the chance to present my research work. So today I am going to deliver the presentation on the effects of orange peel powder on the composition, physicochemical properties, and the sensory quality of frozen yogurt. As you all of know that in case of certain dairy products, most of the knows that the yogurt, which is similar to the dahi, and the second dairy product, if you everyone knows about the ice cream, but this frozen yogurt is the combination of ice cream as well as the dahi. So it is the complex fermented frozen dairy dessert that combines the physical characteristics of ice cream. Means approximately 60 to 70% ice cream was there, with the sensory and nutritional properties of fermented milk product like the yogurt. So if you go for the preparation of frozen yogurt, there might be the ratio of ice cream mix to the yogurt that is vary from 5 to 70%. The frozen yogurt can be made either by fermentation of ice cream mix with the lactic acid bacteria. Generally, in case of frozen yogurt preparation as well as the yogurt preparation, always thermophilic cultures are used, which are Streptococcus thermophilus as well as the Lactobacillus delbrueckii in the ratio of 1:1. So that is called as the direct method, or by the blending of yogurt base with the ice cream mix. So this is nothing but the indirect acidification of frozen yogurt as well as the ice cream mix. Here you can see there are the three classes of frozen yogurt. So on the basis of their fat, these frozen yogurt are classified. The first one is the soft yogurt, the second one is the hard frozen yogurt, and the third one is the moist frozen yogurt. So in case of that soft frozen yogurt, the fat will be vary from 2 to 6, while in case of hard frozen yogurt, the fat again vary from 2 to 6. Only the difference is in case of soft frozen yogurt, the product is stored at below -6 degrees Celsius, while in case of hard frozen yogurt, the product is stored at -23 to -28 degrees Celsius, while in case of this, the third frozen yogurt variety, this can be stored below 0 degrees Celsius. The next one is the naturalness and the health food. As every consumer, there has been the trend towards the food with added natural antioxidant dietary fibers as well as the natural colorants, minerals, and the vitamins. If you know that generally milk is considered as the complete food, but though it is lacking in the certain components, such as it is the poor source of vitamin C, dietary fiber, as well as iron and the antioxidant substance. But suppose if you go for the addition of this frozen yogurt with the orange peel products, mainly that orange peel powder, so these are the resource of that vitamin C as well as the dietary fiber, irons, and the certain antioxidative substance. When you go for the synergy effect, that orange peel powder as the frozen yogurt, so definitely whatever the lacunas are there in the milk as well as the frozen yogurt, we are going to overcome by adding this orange peel powder. Next one is the objective regarding to my research work. In the first phase or the first objective, to assess the suitability of two forms of orange peel products as a flavor adjunct in the medium fat fruit frozen yogurt. So the first one peel form is the candied orange peel, and the second one is the orange peel powder. The second one is to optimize the level of orange peel product and the select appropriate fruit flavoring. So whatever the best in the phase one, I am going to research on that selected form, and in the last phase to characterize the standardized value added medium fat fruit frozen yogurt for their chemical composition, physicochemical properties, and the sensory properties. As you all of know that the orange is the fruit of various citrus species which belongs to the family Rutaceae. Generally, it is originated in the regions of southern China, North India, as well as the Myanmar. In the year of 2019 to 20, approximately 65 million tons of oranges were actually it is already produced worldwide, with the Brazil producing the 24% of the total, followed by the China and India. If you see the composition of oranges, so it approximately contains the 40 to 40% of pulp as well as the juice, 8 to 10% of flavor component, 30% albedo, 20 to 30% of rags, and the 4.4 to that 0% seeds on the total orange. In my study, I am using the orange, Valencia orange, which is generally imported from the Egypt as well as the Africa. So in that fruit, 20 to 28% of that peel, 35% of the juice, and the remaining 25 to 30% is the pomace. So from that peel product, that is the 27 to 28%, I am going to prepare the candied orange peel as well as the orange peel powder, and that both the product I am already using the frozen yogurt. These are the virtues of orange peel, means these are the health benefits. It is the antioxidant as well as it can protect the LDL cholesterol from the oxidation. It also reduces the incidence of heart disease, as well as this orange peel restrict the formation of carcinogenic nitrosamines. The flavonoids such as the hesperidin and the naringenes are the associated with the antioxidant, anti-cancer, as well as the antiviral properties. Here you can see that this is the protocol for the preparation of orange peel powder. Initially, I am just wash the whatever the Valencia orange received. After that, peeling and the cutting stream into these sizes, as well as the next one is the I am go for the blanching. After that, whatever the bittering issues followed in the orange peel that avoid with the help of by dipping in the 2% sodium hydrogen carbonate for the 3 minutes at the 95 degrees Celsius. After that, these orange peels, de-bittered orange peels, are go for the sugar syrup treatment at the 50 degree bricks for 4 hours at the 50 degrees Celsius. After the 5 hours, just drain the excess sugar syrup. Again, the vacuum tray drying at 45 plus minus 2 degrees Celsius for the 8 to 10 hours. It is depend upon. So the final moisture is less than 8%. After that, milling and sieving was finished, and the whatever the orange peel powders with the 8%, 7 to 8% moisture, just pack in the airtight jar and the stored at below 6 degrees Celsius. This is the preparation of frozen yogurt containing the orange peel solids. First of all, I am weighing the ingredients. In the mixed formulations, I am prepared the 4 batches. The first one, that is the control, in which there is no addition of orange peel powder, and the next 3, these are the experimental frozen yogurt. The fat and SNF as well as the sucrose content and the stabilizer are the same. Only I am using 1.5% orange peel powder, 2.5% orange peel powder, and the 3.5% orange peel powder. After the mixed formulation, that homogenization was carried out so that there is no fat layer observed. Then the pasteurization at this 80 degrees Celsius to kill the pathogenic microorganisms. This cooling and aging at 4 degrees Celsius. So definitely this aging is help to increase the viscosity of mix as well as for the proper foaming properties. Then after the aging, I'm going to add flavoring, that is the orange crush at the 9% as well as the freezing carried out at -5 degrees Celsius. Before freezing, I'm added the yogurt. After that freezing, packaging and the hardening in the blast tunnel. So generally this hardening is carried out at -23 to -28 degrees Celsius for 3 hours, and after that store the product at -18 degrees Celsius plus -2. Here these are the orange peel products. These two represent the candied orange peel. I am going to add by this slicing form, and in the third picture you can see that orange peel, though the name is orange, but the color of this orange peel is slightly yellowish. Now the results, whatever the results here in this table, you can table 1, you can see that the proximate composition of frozen yogurts prepared by using varying level of orange peel powder. So add the fat content is the non-significant. Definitely the fat percentage I am keep the same throughout the product, but this protein carbohydrate is vary. It is depend on which type of milk we are going to use for the standardization, and as well as the this 1.5 to 2.5 and the 3.5% OPP. So definitely as the percentage of orange peel powder is increases, it definitely increase in the protein content, as this orange peel powder consists about the 5.7% protein, as well as this carbohydrate is also increased, and ash is also increased, and the TS is also increased. As the level of this orange peel powder increases, definitely there are the increase in the protein, carbohydrate, ash, as well as the TS of the product. So these are the composition. Here in this table, you can see that the physicochemical properties of frozen yogurts. In case of certain dairy products, namely that ice cream as well as the certain frozen dairy desserts, this physicochemical properties, mainly viscosity as well as the overrun and the melting resistance, is very important. So here in this case, you can see that the acidity increases from 0.46 to 0.5% lactic acid, while in case of this control, the acidity is lower. Definitely this orange peel are slightly acidic in nature, as due to the contribution of citric acid. So that's why there is increase in the acidity. Now the viscosity is very important parameters. Viscosity of mix, here you can see that the viscosity of mix is 149.06 centipoise, while in case of this experimental frozen yogurt, definitely the viscosity is more than 2 or the 2.2 times. So this is due to the addition of orange peel powder. If you see the standards for the viscosity of ice cream mix, so there is the range of 50 to 300 centipoise, but definitely this initial pectin content of orange peel powder, it will lead to the viscosity of this experimental frozen yogurt mixes. So as the level of orange peel powder is increases, the viscosity is increases. Overrun is also, generally this overrun is nothing but the additional amount of that frozen yogurt in the volume as compared to the initial weight of frozen yogurt mix. So this in case of the control frozen yogurt, there is the higher overrun. As you are going to increase the OPP, there is the decrease in the overrun. So generally, if you see, this is the drawback of frozen yogurt quality, but definitely it will increase in the viscosity as well as the melting resistance. After that, I am going to discuss what is the effect of this orange peel powder, varying level of orange peel powders on the sensory, because on the basis of sensory, whatever the product is finalized, we are going to compare with the control in the latter phases. Here you can see that the sensory scores of frozen yogurts, varying level of the orange peel powder. So among that 4 treatments, as you all know that the first one is the control without the use of orange peel powder, while in case of this 1.5, 2.5 and 3.5 OPP, you can see that as the OPP is increases from 1.5 to 2.5 or 0 to 2.5%, definitely there is the increase in the flavor score, and after that slight decrease in the flavor score. This may be due to that the flavor intensity is slightly high in case of this 3.5 OPP, body and texture is also the similar trend. At 2.5%, the body and texture is the good, while as the rate of increase is 3.5%, definitely the body and texture score will reduce, because as per the judges, they feel the certain chalkiness effect as well as the powdery effect. That's why the higher rate of OPP does not score the as much as this 2.5% OPP. In this again, the melting quality. Actually, this melting quality is very good in the 3.5 OPP. The color is definitely non-significant, because the effect of this OPP is not the as differ as compared to the control as well as the OPP. Definitely here in this total score, the highest score is observed for the recorded for the 2.5 OPP frozen yogurt, as compared to, while in case of this 3.5 OPP, it is the lowest score. Even it is lowest as compared to the control also. Here in this graph, you can see that the effect of OPP on the basis of OPP in the frozen yogurt. So the values represent in the green color definitely are the higher, which represent the 2.5 OPP. So from this graph, you can conclude that the flavor as well as body. Yes, sir.
Kindly wind up.
Yes, sir. Was last slide conclusion.
Sure.
Okay. And now I'm going to conclude my session. So definitely, as we see, we saw that as the level of OP increases, there is the significant increase in the protein, carbohydrate, ash, and the TS of the product, as well as this OPP significantly affect the acidity, viscosity, O-run, and the melting rate of the frozen yogurt. Frozen yogurt containing 3.5% had the highest acidity and the viscosity, but this 3.0 OPP is not optimized. Only the results of the study revealed that the frozen yogurt produced with 2.5% OPP result in the orange flavored frozen yogurt that was significantly superior as compared to other three treatments. So thank you. Here I'm just complete my presentations. If any queries, then most welcome.
Yes, thank you, Sheikh Agni. It was
Yes,
a
ma'am.
very well presented flow of your results. We have one question from Dr. Alka Devi. She
Yes, ma'am.
asked,
Okay.
is there any effect on probiotic nature when the orange peel is added?
Yes, ma'am. Actually, as per the FAO, generally, if you say the any product 10 to the power 7 CFU, so this orange peel powder is act as the prebiotic. So definitely it increases the viability, as well as up to the 6 month or 4 to 6 month storage, it will maintain that viability throughout the storage period.
I think what she meant is the probiotic microbes. I hope.
Yes, ma'am, definitely. These cultures will also act as the probiotic, this Streptococcus thermophilus. And if you add the probiotic cultures, this OPP will increase its probiotic count, or at least as compared to the control product.
Okay, you mean to say that OPP is supplementing the food source for the growth of bacteria?
Yes, ma'am.
And what was the acidity levels once you added OPP and the sugar powder, orange peel? Is there a difference?
Yes, ma'am. Actually, here I am mentioned.
I can-
Actually, the acidity is 0.46 to 0.51. I am take the 75% ice cream mix with the acidity of approximately 0.35 and 25% of yogurt mix with the acidity of 0.55 to 6. So definitely after the blending of these two mixes, the final acidity in the range of this 0.46 to 0.51% lactic acidity.
Okay, fine. And is that palatable for a human? Matlab, we can take it without harming ourselves, such an acidic kind of background solution?
No, yes, ma'am. Actually, certain fermented products, if you see the yogurt as well as the Srikhand, their acidity is up to 1.4%.
Yeah.
So this is the mild acidic.
Okay, fine. Thank
And
you.
for frozen yogurt, the minimum acidity should be 0.35%.
Okay. Okay, fine. Thank you, Sheikh Adil. Thank you.
Okay, thank you, ma'am.
You
Okay.
can unshare your screen. May I now
Yes,
request
yes,
the
sure.
next participant, Miss Neeta Kaushik? Miss Neeta Kaushik?
Yes, ma'am.
Yeah. As soon as he unshares your screen, you can share and start with your presentation.
Okay, ma'am. Okay. Ma'am, my screen is visible for you.
It's showing started screen sharing. So we can wait for 1 minute.
Okay.
It's not visible as of yet on our end. Yeah, it's visible. Now you can put it on full screen and start.
Now it's visible.
Full screen,
Okay.
please.
Can I start, ma'am?
Full screen, please. F5.
Full screen. Okay, ma'am. Now,
And kindly go to this slideshow option. Home.
click on the.
Yeah.
Now is it visible on full screen?
No. You have to start the slideshow. Now you are unshared, I think.
I'm trying to share it.
Please share it.
Sir, is it visible?
Just wait for 5-10 seconds.
Okay. So can I start now?
Please wait. We are unable to see the screen. Okay, now it's visible. Kindly put it in slide share mode or slideshow mode.
Okay.
Close to the this bottom right corner, you have that close to the window. Yeah.
Yes, I clicked on that, sir. Actually, my internet connection is unstable.
Okay, fine. You can start.
You might,
with it.
sir. Let's
You
not
can
wait.
start.
Thank you, ma'am. Thank you, sir. So, very good morning to all present here. I, Neeta, research scholar from Guru Jambheshwar University. Today, I will present on advanced spoilage detection methods for fruits and vegetables. We all know that Indian population is mostly vegetarian. So basically, fruits and vegetables are an important part of our daily diet. And that's why India is the fourth largest supplier and second largest producer of vegetables in the world.
Neeta, are you there?
Sir, I am here.
Your screen is unshared.
Actually, there is a dialog box which representing that your host muted everyone.
No. Let's start.
Okay. So, as per an estimate, fruits and vegetables are lost every year due to spoilage, and around 1 tons of horticulture commodities are lost and squandered each year, which accounts for 30% of output made for human consumption. So, major cause is spoilage in loss of horticulture commodities. When the spoilage occurs in fruits and vegetables, it makes them unedible and unfit for human consumption. It changes the quality of fruits and vegetables by changing the color, flavor, texture, and aroma. The spoilage mainly occurs during the post-harvest handling and post-harvest storage. So basically, there are the major three factors which are responsible for the spoilage. First are physical factors. Physical factors which affect the quality of fruits and vegetables, such as temperature, moisture, pressure. Overall, we can say that storage conditions which are responsible for causing the spoilage, and which are also caused by the freezing, burning, drying, and bruising of fruits and vegetables during the storage, handling, and transportation, which results in the food deterioration. And in case of chemical factors or chemical reactions, which involve the exposure to oxygen and light, which are the two most common causes of spoilage. Oxygen can cause spoilage in several ways, like encouraging the growth of microorganisms, resulting in mold and yeast growth, and also oxidizing enzymes, which are responsible for speeding up the chemical reactions in food, resulting in the browning and fall odors. So, for example, we can say that tomatoes become soft, apples turn brown due to the browning reactions, and due to the oxidation, fruits and vegetables become rancid and totally unfit for human consumption. And the third one, microbial factors, which are the major cause of spoilage, especially in India. These causes mainly occurs during the post-harvest storage. In fruits and vegetables, spoilage starts with hydrolysis of pectin. Once the pectin is damaged the structure, pectinase is an enzyme which responsible for damaging the structure of fruits and vegetables. And at the same time, other microorganisms start to contribute soft rots. You can see here the microbial spoilage pictures of fruits and vegetables. The major microbial spoilage are saprophytes, pseudomonas, polyhomes, and many more, which may be from soil and water. In the fungus, in case of fungus, we can have Fusarium and Alternaria are responsible for the microbial spoilage. And in this table, you can see that basically the spoilage occurs in several series of rots, like bacterial soft rot, gray mold rot, rhizopus soft rot, and the causative organisms are like Erwinia, Botrytis, Rhizopus, Colectotrichum, Alternaria, and many more. And in case of effects, they totally affect the texture, aroma, and the structure, and also the taste and odor of the fruits and vegetables. And if we discuss about the microbial spoilage scenario in India, we can say that we are very well known that India is the world's second largest producer of fruits and vegetables, which ranks next to Brazil and China, and it also contributes about 10% of global food production and 14% of global vegetable production. Here you can see that in India, wastage of fruits and vegetables, which mainly occurs in potatoes, apples, and onions. And it mainly occurs during the post-harvest storage handling practices. Now we discuss about the spoilage detection techniques. So here I want to tell what is the need of the spoilage detection. Because testing for the earliest spoilage detection, which is used to examine and so that we can prevent the food poisoning outbreaks which are caused by the food products and ingredients and microorganisms. So here are the spoilage detection techniques, mainly biosensors, electronic NOS, electronic drum, and smartphone-based technologies. So in case of biosensors, biosensors are the sensing devices which comprise specific biological element and a transducer. A specific biological element called bioreceptor, which generally recognize the specific analyte and changes in the biomolecular structure and usually convert it into electrical signal by a transducer. Transducer is another component of biosensor which generally used to convert one type of energy into the another. Here, the biosensor detection scheme. Analyte recognized by the recognition elements and the transducer, which are generally used to convert the form of energy from one electrical signal fast, and we can easily detect the spoilage through the biosensor detection scheme. And this electronic sensor, electronic the sensor system mainly comprised of three parts: the system, sensors, it is basically used for the detection of the volatile compounds, and it is used in the major applications like in tea, wine, coffee, and spices industry. So here is the electronic transistor. Next is electronic tongue sensor. It is also a kind of sensor, biosensor, which is a novel technique used for the taste evaluation of foods. Multisensory framework consists large number of sensors and uses advanced mathematical procedures for signal processing based on the pattern recognition or multivariate data analysis. It is used in the food items to categorize, authenticate, quality evaluation, process monitoring, and quantitative analysis of foods. Basically, here I want to tell you that electronic nose and electronic tongue is a combination of gas sensors and chemical sensors, which mimics human nose and human tongue. So, electronic nose are gas sensor arrays, while electronic tongue are chemical sensor arrays. Here, if you put the sample into the sensory chamber, then chemical sensors evaluate the sample, and they feature the extractions and will pass the signal into the computer system, which will recognize the pattern, and information will be displayed on the display screen. And next one is smartphone based techniques. Smartphones, which are very convenient and as powerful as computers, which are widely used in biosensing assays, acting as a detectors, data processors, and signal inducers. Why we use smartphones in techniques? Because they are more convenient, more accessible, fast, and cheaper than portable analytical laboratory devices. So mainly smartphones are combined with the microbiosensors, microelectronic devices, microchips together to achieve real-time point of care and easy to use predictions for things. So here we use smartphone-based technologies in each and every kind of commodities rather than fruits and vegetables. So here now I'm going to on my conclusion. This review outlined the advanced spoilage detection methods for fruits and vegetables, such as electronic nose, electronic tongue, biosensors, and smartphone-based technologies are some of the promising trends of spoilage detection. In future, use of these techniques on wide scale would be a new trend of spoilage detection, not only fruit vegetables, but including all other commodities and various food products, which can improve the microbiological quality while lowest the impact on the freshness, organoleptic properties, and quality of foods. And the use of chemical biological sensors and indicator labels in active and smart packaging, the advanced approaches which can be used to evaluate the quality of fruits, vegetables, or food products is a growing area of research, and the most effective sensors to date have been used to detect volatiles such as amines and ethyl. So future advancements must focus on lowering the limit of detection and raising the ability to compute on single contacting with the material. So thank you so much.
Thank you, Neeta. May I have some questions from the participants that you may like to ask Ms. Neeta Kaushik? Is anybody trying to ask any question? I guess not. Okay, in that case, Neeta, could you tell me the names of the chemicals the electric tongue would be detecting?
Used to the food quality control to measure the amino acids, alcohols, procyclic compounds.
Which amino acids may I ask? Which amino acid is an indicator of spoilage? I think she is no more in the session. Yeah, she's back. Hello, Neeta.
Am I audible?
Yes, you are audible. Could you hear my question?
Yes, ma'am, exactly. Ma'am, actually, in case of electronic tongue, these are used in food quality control, especially to measure the. Am I audible?
Yes, you are audible.
Okay.
Yeah, but you didn't answer the question. I said which amino acid is an indicator of spoilage? You said amino acids are being detected by the electric town. So which amino acid is an indicator for spoilage? Can you tell me that?
Actually, it is used to measure the amino acids like amides, amines, carbohydrates, carboxylic acids.
Amides, amines are not amino acids. There is a specific amino acid which is an indicator of spoilage.
Ma'am, actually, I have no idea the particular amino acid.
And you had also mentioned some VOCs. You know what are VOCs?
What, ma'am? Sorry.
OCs, volatiles, volatile organic compounds. Can you name some volatile organic compounds the electric nose will be detecting?
Ma'am, actually, they used to detect the natural toxins,
Toxins will
which
be
we can't. Yes, ma'am.
they will be detected by the tongue. Anyways, thank you, Neeta. You can.
Thank you so much, ma'am.
Okay, the last. Okay, the second last participant for this session is Miss Hina Bisht. May I request her to upload her slide? Hina Bisht.
Good afternoon, ma'am.
Yeah,
Am I audible?
yeah, you are audible. You may please share your screen.
Okay. So, good afternoon, everyone. So, my name is Hina Bisht, and I'm a research scholar in NIT University. So, the topic of my presentation today is isolation and identification of aluminum tolerant bacteria. As we all know, over the last few decades, due to increase in population, urbanization, and industrialization, problem of environmental pollution is escalating day by day, and which contaminate our soil, sediments, groundwater, and surface water. So there are lots of pollutants in our environment, but the most menacing ones are the metals. So there are many metals which contaminate our environment, like nickel, lead, chromium, etc. But the metal which I am discussing today is aluminium. So what are the sources of aluminium metal? So first is acidification of the soil by mining, aluminium industries, you can say aerospace industries and transportation industries, and also in food additives and packages and containers, flocculants, and also in drugs and cosmetic industry, they have been using aluminium. So with the help of, so they discharges the aluminium in a soil and the in a rivers without treating it. So what are the toxic effects of aluminium in plants and in humans? So in plants, it inhibits the uptake of many cations such as calcium, magnesium, and potassium ions from the soil, and also inhibit the root growth, cell division, and also reduces stomatal opening and photosynthetic efficiency. And in humans, it is extremely neurotoxic, and it causes many neurodegenerative diseases like Alzheimer's and Parkinson's disease. It also causes various bone abnormalities and also pulmonary fibrosis and also gastrointestinal problems. So there are many physicochemical recovery methods also which help in to decontaminate the metals. But the bioremediation methods are the most good ones because in physicochemical methods they are mostly costly, and sometimes it generate most toxic sludge. So in bioremediation, many aluminium tolerant fungi include Saccharomyces, Lipomyces, Rhodotorula, and ectomycorrhizal fungi has been reported by many researchers, but a very few studies on aluminium tolerant bacteria. So some bacteria like Flavobacterium, Pseudomonas, and Arthrobacteria, Bacillus cereus, etc. were there. So in our studies, we are trying to isolate a bacteria which can tolerate more of the metal ions, like aluminium. So First of all, isolation of metal tolerant bacteria. So we have collected the soil samples from the disposal of metal contaminate waste in the vicinity of metal electroplating and automobile workshops. So after then, the bacterial isolates were screened on luria agar plates supplemented with fixed concentration of aluminum metals. And the morphological dissimilar colonies were randomly selected, and we got the 7 metal resistant. After screening methods, we got the 7 metal resistant cultures named as M1 to M7. After that, we have grown in the fixed concentration of aluminum metal. So this is a graph. So the growth of M1 is more as compared to the other isolates. So we have selected the M1 isolate for further research. For the identification of bacteria, first of all, we have done the biochemical analysis. So in biochemical analysis, we have done the ground staining, starch hydrolysis test, oxidase test, gelatin hydrolysis test, citrate, and catalase, and many more. So after that, we have done the 60, after biochemical analysis, we have done the 16S rDNA amplification. So for this, we have first isolate a chromosomal DNA of isolate M1. So this is the picture. And after that, we have done the 16S rDNA amplification. So it was done by using universal primers 8F and 1492R. So we have observed the 1.5 kb band, that is 16S RNA DNA band. After then, we have done the sequencing. Sequencing was done, and we have sent the sample to the Amnion Biosciences, Bangalore. And the result of sequences were compared with the existing sequence in GenBank database using NCBI BLAST. So after analyzing, we have concluded that our isolate M1 was 99% similar to Sedtia davisi isolate, which is from the group of Enterobacteriaceae family. So we have also submitted in the GenBank, and this is its accession number. After that, we have done the maximum tolerance concentration, that how much this bacteria tolerate aluminium metal. So for this, we have grown it in different concentration of aluminium metal. First of all, 50, 75, 100 mg per liter, then 150, and then 200. And also without metal also. After that, we have incubated at 37 degrees centigrade at 150 RPM. And then measured the OD at different intervals of time, like 0 hour, 12 hour, 24, 48, and 72 hours, with the help of UV visible spectrophotometer at 600 nanometer. So here's the graph. So at the topmost, this is our isolate, our Sedtia davisi M1 without metal. So it grows maximum. After that, this is blue line. In it, we have put 50 ppm aluminium. So as the concentration of aluminium increases, the growth rate decreases. So that means the tolerance level also decreases. So at 200 ppm concentration, the growth rate decreases drastically. So we can conclude that the maximum tolerance level of aluminium for the bacteria is 150 ppm. So with this, we can see that there are some genes or some mechanism that is responsible for the tolerance of aluminium in Sedtia davisi M1. So this is our future studies. We will find it out, and after that, we will analyze the bioremovable efficiency of aluminium by Sedtia davisi with the help of atomic absorption spectrophotometer. So conclusion, as our isolate Sedtia davisi M1 tolerate high degree of tolerance to aluminium metal, it can act as a potential and novel candidate for the bioremediation of aluminium from the contaminated sites in future. Thank you.
Thank you, Hina.
Yeah, ma'am.
Are there any questions from the participants? Okay. In that case, Hina,
Yes, ma'am.
all a very good initiative.
Thank you.
One thing I would like to ask, why have you named it aluminium tolerant?
Ma'am, because
Tolerant bacteria does not help, right?
yeah,
So why an aluminium tolerant when your ultimate aim is different?
Ma'am, first is tolerance. We have to just analyze the tolerance level, how much it tolerate, that at what ppm concentration, what concentration of aluminum it can tolerate, or it can grow at what concentration of aluminum. After that, we will find out the mechanism, whether it absorb or accumulate or efflux the metal.
So the efflux will again lead it back to the soil, right?
Yes, ma'am.
Okay. Then you said you have 7 isolates, right? But I
Yes,
could
ma'am.
feel the RRDNA, ये तुम्हारा ribosomal DNA for only 1 isolate.
Yes, ma'am. Yes, ma'am.
Find that all the 7 were belonging to the same species.
No, ma'am, different.
Then how did you label it in the graph that all of them are Sedixia?
Ma'am, actually, I have isolated the 7 isolate, and I'm also working on different other metals also. So the like nickel, cadmium also. So the aluminium metal which grow. So in isolate M1, it grow more as compared to other metals. So for aluminium, I have taken this isolate. For other metals, I have taken the other isolates.
Okay. So when you have shown the graph, it is at the different concentrations. Is that it?
Yes, ma'am. Yes, ma'am.
Okay. So the sustainability of your culture is maximal at which concentration as per your result?
Ma'am, it grows at 24 hours. It grows after that, it shows the stationary phase. So at 24 hours, it grows maximum.
Concentration, ppm.
The ppm is 150 ppm.
But the green, okay. 150 कहाँ से हो गया? Yellow.
Ma'am, this is yellow is 150 ppm. After that, the last one is 200.
Highest I asked, which
Yes.
is the highest tolerance level of the bacteria I asked.
150 ppm, ma'am.
150 नहीं है ना बच्चे. Blue वाला सबसे highest दिखा रहा है ना आपको?
Okay, ma'am, that is the highest. Okay, ma'am, that is 50 ppm.
Yeah, thank you. Thank you, Hina.
Thank you so much, ma'am.
Okay. May I request the next participant, Pooja Chavra? Pooja Chavra?
Yes, ma'am.
Yeah, please share your screen and initiate your presentation.
Okay. Ma'am, is it visible?
It's visible, yes.
May I
Yes,
start, ma'am?
yes, yes.
Good afternoon, everyone. Welcome you all. My name is Pooja Chabra, and I am a student pursuing PhD from Baba Masnath University, Rohtak. Today, I am going to talk on use of SORT-Me RNA tool to mine the diverse RNA molecules from buffalo milk transcriptome. I will begin my talk by outlining the rationale behind choosing this topic, and then I will highlight the steps of algorithm that is being used by the SORT-Me RNA tool to separate the rRNA fragments from the transcriptomic data. And finally, I will briefly discuss about the steps that are applied to the SORT-Me RNA tool output to mine the diverse RNA molecules. Now I will focus on the rationale behind the topic. The RNA molecules that are not coding to any protein are known as a non-coding RNA molecules. These are rapidly becoming more important as diagnostic and treatment tools for any disease relating to physiology, genetics, epigenetics, and developmental defects. There are various data mining tools that can be used to extract these RNAs. This study is the first attempt to mine the different kinds of RNA molecules from RNA-seq data using SORT-Me RNA tool. Now, I will discuss the central dogma of molecular biology. The genetic information of all the living organism is stored in the molecule known as the DNA, and all the DNA molecules have the double helix structures. And this double helix structure is represented by the two strands that are running in the opposite direction, represented by the 5N and the 3N prime. And these two strands are composed of the units which are known as the nucleotides that are distinguished by the nucleotide bases ATCG. And these two strands are also paired or bound together by the corresponding nucleobases, that is A always bound to T and the C always bound to G. With the help of an enzyme, DNA is disintegrated to form the RNA, that is a single-stranded, and further the RNA is disintegrated to form the protein. This is the representation of the next generation sequencer output, where the DNA is represented by the 2 strands, and this is represented in the form of the ATCG sequences. With the process of transcription, DNA is converted to RNA, which is a single-strand molecule, and all the T's are represented by the all the T's are replaced by the uracil U. Further, the RNA is translated to form the protein. In the RNA, the combination of the three nucleobases, that is just like AUG, this is known as a codon, and each codon is corresponding to a particular protein. There are this is the role of the diverse RNAs. If we want to know the functional elements of the genome, then it is essential to know the functions of the gene, and these RNAs determine the function of the gene. So hence it is important to know what are the roles of the different RNAs. In the slides, it is given that it is shown clearly that snRNA regulates the RNA splicing, whereas the snRNA and mRNA participates in mRNA splicing. MicroRNA involves in translation repression, whereas the piRNA controls the gene expression during transcription and post-transcription processes. LincRNA plays an important role in epigenetic regulation. rRNA regulates RNA splicing, and there are various data mining tools that play an information about all these RNAs. Now we focus on the data mining. Next generation sequencer produces a plethora of the biological data, and the domains that are leveraging with a rich amount of data are the best candidates for the data mining. Hence, enhancing the communication between the NGS data and the data mining techniques has a great potential. With the rapid growth of NGS application, new methods of data storage, analysis, and visualization are required. Data mining is a term that refers to obtain the knowledge from the large amounts of data by digging or mining. It's a process used to extract usable data from a larger set of any raw data. It is a technique of discovering interesting and useful patterns and relationships in the large volumes of data. Now, I will describe the methodology to extract the different kinds of RNA that I have used. First step in this method is the data collection, then data pre-processing, then processing with the SORT-me RNA tool, then mapping with genome, and the last step is mining of the different RNAs. Firstly, I would like to describe what is a SORT-me RNA tool. That tool is specially designed to separate the rRNA data from the transcriptomic data. It is capable of finding the large quantities of reads and sorting them out with a high speed by looking for the set match the rRNA database. It takes FASTA or FASTQ for input and one or more rRNA databases, sorting out the aligned and rejected reads into 2 independent files. It comes with the 8 representative rRNA databases. It is based on similarity searches between an RNA database and a read. Now I will describe the data mining steps that are used for mining the different types of RNAs. First step is data collection. SRA at NCBI is the primary source for the next generation sequencing data. And this work adopted a real data set which was downloaded under the bioproject number BRJNA453843. 12 FASTQ files of buffalo milk somatic cells were downloaded. And next step is data pre-processing. As a result of the experimental and the sequential processing, the raw data contains the biases and the complex artifacts. These artifacts strongly influence the accuracy of the read alignment and the subsequent analysis. So to reduce the computation requirement and to increase the downstream analysis accuracies, it is best to pre-process the data. For checking the quality statistics of the raw samples, VastQC tools was used, and this tool provides us the information like the GC content and the number of reads, length of the reads, whether the data sequences are present in the raw reads or not, or the overrepresented sequences. This are based on this information that was provided by the FastQC tool. And next, I have to apply some filters, like the length of the reads was 1 nucleotide to 101 nucleotide. But whereas my focus on mining the RNAs where the length is greater than 20 nucleotides. So there was no need to maintain the reads whose length is 20 nucleotides. So I have to apply the filter that all the reads less than the 20 nucleotides have to be removed. So I applied the length filter. Next, there were adapter sequences also in the raw data. I have to also remove all these adapter sequences. Next, there were the reads whose quality was very low. I have put the quality threshold that is 30. Means all the reads whose quality was less than 30 or Phred score was less than 30 was removed. All these filters are applied by using the tool that is Cut-A-Wrap. Now processing with the SORT-me RNA tool. So this is a algorithm that was used by the SORT-me RNA tool. In this, the RNA database is stored in the form of the bus style, and which is coupled with the lookup table to speed the access to that bus style. With the given read and the windows of the read, we can find the multiple windows in the bus style using the universal Levenshtein automata. This comparison is done parallel with the universal Levenshtein automata and the bus style. It means that we have given a read and we are taking the multiple windows of that read. If that window, we will check the prefix or the suffix, whether prefix or suffix is in the lookup table. If it is there, then we will go to the further processing. Otherwise, we will reject that window. If the prefix or the suffix is there and the universal Levenshtein automata generates a word, generates a different or the same word with at most one error from the bus style, then we'll accept that window. Otherwise, we will reject that window and we will go to the next window to check the matching with the RNA database. If the sufficient number of windows are accepted for that read, then that tool has classified that read as a rRNA. Otherwise, it will be rejected as an rRNA. This is the algorithm that I have explained. The lookup table stores all of the SMERs and their number of occurrences that exist in the rRNA database. The algorithm takes as input a collection of reads provided by the user, and for each read, a sliding window W of even length as moves across the read. For each window W, the prefix and the suffix are translated into a decimal value between 0 and 2 raised to power s minus 1. If the value exists in the lookup table with a high frequency, the remaining part of the window is searched in the rRNA database. After all windows have been traversed, if the number of accepted window exceeds a certain threshold, then the read is accepted and classified as rRNA. At the end, it generates the two files, RNA accepted file and RNA rejected file. Now, I have two files, that is RNA accepted and RNA rejected. After getting these two files, the next step is mapping with the genome. Mapping the generated files was done with a human reference genome using an aligner. I have used BWA aligner. And next step is the mining of different RNAs. The mapped files generated by the aligner were converted to transcripts file using the tool couplings. The generated transcript file was filtered on the basis of the different RNAs. These are the databases that were used by the SORT-me RNA tool, because the SORT-me RNA tool is only separating the rRNA sequence. So it uses only the two databases, that is SILVA and RFEM, that is based on the rRNA sequences. These are the results that I have got, and this graph is showing the distribution of raw reads versus processed reads across the different samples in buffalo milk transcriptome. The raw sample had about 47 million reads in each sample, and about 45 million reads were obtained after filtering and trimming from each sample, and about 30 million reads were trimmed due to the reasons such as the short length, adapter sequences, or the poor quality. Next, the processed reads were mapped with the human reference genome because it is better annotated at the buffalo genome. So this graph is showing the alignment of the processed reads with the human reference genome, and it is showing that all the reads, all the samples are showing the highest rank mapping with the human genome, ranging from 85% to 99%. This is a summary of the different RNAs mined from buffalo milk somatic cell samples. If we want to know the functionality of the genome, then it is compulsory to know the function of the gene, and different RNAs determine the functions of the genome. So the quantification of RNA is mandatory as they imply the cause of chronic diseases. And I have put the link RNA in my sample with the highest count, that is 2,444, followed by the snRNA and snRNA, which were 58, then microRNA, which were 20, rRNA 31, and tRNA 4. However, there was no pyra in my data, but it was compared. Now, it is time to sum up the information. Here are some key points. We were able to achieve the microRNAs, pyRNAs, tRNAs, snRNAs, snORNAs, rRNAs, and link RNAs in buffalo milk somatic cell transcriptome. Maximum number of link RNAs were identified in our data set, followed by the snRNAs and snORNAs. SORT-me RNA tool output can not only be used for separating the rRNA fragments, but also for the detection of the other known coding RNAs like microRNAs, tRNA, link RNA, pyRNA, snRNA, and snORNA. Now it's time that's all that I wanted to say. Now the session is open for discussion. If anyone has any question, then I will really be glad to answer. Thank you.
Thank you, Ms. Pooja. An excellent
Yes,
presentation
ma'am.
and a very viable study. But yeah, what will be the ultimate aim of your study? Why do you want to identify all the constituting, or rather not constituting, the RNAs present in buffalo milk?
Actually, ma'am, if we want to know the functionality of the genome, then the functionality of the genes can be determined from the RNAs. And coding RNAs are already being researched. There are the known coding RNAs where the research is not being yet. So my focus is on determining what are the known coding, how many known coding RNAs are present in the data, so that we can annotate the buffalo genome, because the buffalo genome is not yet completed.
So why milk as in sample?
Yeah, milk, by discovering all this RNA, milk production of the buffalo can also be increased. If we know the count of the RNA, that is highly influencing the milk quality.
So
But before, yes, ma'am.
are you correlating the non-coding RNAs with the milk production? Is there a correlation study being performed?
Yes, ma'am. There is an effect of this non-coding RNA to the milk production. Yes, a study has been done.
Okay, fine.
So before, yes, ma'am.
Thank you. Thank you so much. Thank you all the participants for patiently giving their presentations, and all of you have given an excellent presentation, and thank you so much.
Thank you.
Okay. Thank you, all the participants, for your time. I would also like to take this opportunity to thank our session chair, Dr. Manali Datta. She has taken a time out from his busy schedule and be available for this conference. So I thank you all once again.
Thank you.